Here we have coins representing the possible phenotypes of a species of turtle in the wild. Adult size is dependent on an autosomal dominant gene. Small individuals are homozygous recessive, medium individuals are heterozygous, and large individuals are homozygous dominant. Let us say that I went out to the wild and collected a sample of 100 adult turtles from the thousands that inhabit the ecosystem. Since their adult size is dependent on genotype, we can tell these turtles' genotypes based on their size. These 5 are homozygous recessive, these 34 are heterozygous, and these 61 are homozygous dominant. If the number of genotypes in the population match the number of expected genotypes based on allele frequency, then the population is said to be in Hardy-Weinberg equilibrium. First, we must find the allele frequencies, then use the Hardy-Weinberg equation to solve for the expected number of genotypes in the population. Using a chi-square test, we can statistically check if the number of individuals that we observed is close enough to the number we would expect to consider the population in Hardy-Weinberg equilibrium. Since our chi-squared value is 0.01, which is much smaller than our critical value, we can assume that the population is in Hardy-Weinberg equilibrium. Let's imagine that the migratory route of a species of predatory bird has changed such that it now completely overlaps with the population range of our turtle species. As these birds pass through the turtle territory, they will feed on the turtles, but some phenotypes will be more vulnerable than others. The large turtles are too big for the birds to grab with their talons, and the small turtles are too small to be seen by the birds while in flight. Only the medium-sized turtles are small enough to be picked up and large enough to be seen. This change in relative survival rate is called fitness and creates a selective pressure. The fitness is essentially a multiplier that modifies genotypic frequency. The fitness of the large and small turtles will be 1 since they are unaffected, but the medium turtles will have a fitness of 0.15. This means that once selection has taken place, the genotypic frequencies in our population will change. This type of selection, in which homozygotes are favored, is called disruptive selection. Immediately after the migratory birds pass through the turtle's territory, we sample 100 more. This time we have 7 small, 7 medium, and 86 large. Clearly we can see that the selection had a massive impact on the medium-sized turtles as their representation in our sample has decreased by 27%. But why did the other groups increase if only the medium-sized turtles were affected? Well, we lost 27 medium-sized turtles, but our sample size is still 100 turtles, so those missing turtles must come from the other groups. The homozygous dominant group increased more than the recessive group because the dominant allele has a higher frequency than the recessive one. So, can we mathematically demonstrate that this selection has violated the Hardy-Weinberg equilibrium? We can repeat the same process as before, finding the allelic frequencies and using the Hardy-Weinberg equation to calculate the expected genotypic frequencies. We then use the chi-squared test to check the statistic significance. Since the chi-squared value is 39.48, much larger than the critical value, we must reject our hypothesis. This population is not currently in Hardy-Weinberg equilibrium. If this selection continues, we expect to see the frequency of the dominant G allele overpower the recessive one. After only a few generations and rounds of selection, the remaining heterozygous and homozygous recessive individuals will die out.